So we did a study on the gut microbiome. A microbiome is a group of microorganisms that inhabit an environment, and the gut microbiome is the bacteria that inhabit the gut regions of our bodies. They have been proven to be linked to psychological conditions. For example, a study was conducted by researchers on 1,054 humans in the Flemish Gut Flora Project. It was found that gut bacteria produce compounds that act on the nervous system. One thing found was that Corporococcus and Lister were linked to mental illness, being more common in people without depression because of their ability to produce DOPAC. This was a correlative relationship found in the study, along with the other types of bacteria that are found to be negatively or positively correlative to mental health. Another study was conducted by neurologists on Parkinson's disease, and it was found that the abundance of proteolactate in feces of Parkinson's disease patients was reduced by 77.6% as compared with controls. The relative abundance of Enterobacteriaceae was positively correlated with postural instability and gait difficulty, which are results of Parkinson's disease. So our question was, will similar weight have an effect on the gut microbiome? And our hypothesis was that the gut microbiome would be the same. So in our study, we compared 4 animals. 2 samples were huskies and 2 samples were pugs. All 4 types were dogs. The huskies were both females and the pugs were both males. Both of the pugs were 2 years and 2 months old, but the huskies, Maya was 3 months and Nori was 2 years and 5 months old. Nori and Buster both weighed 37 pounds and Maya weighed 19.1 pounds, while Cooper weighed 35 pounds. The only dog that had health issues was Maya. She had Caucadia. She was also the only dog that stayed indoors. The other dogs went outdoors and indoors. Both of the huskies exercised a lot, and both of the pugs exercised occasionally. Cooper and Buster had only eaten neutral ultra for their entire lives, and the huskies' diets vary from each other. Nori had a raw diet for 1.5 years, and Maya had a grain-free diet for 2 months. By the end of our project, we had changed our samples to two samples of 2 flat-coated retrievers, and their names were Husky and Nova, which both weighed 50 pounds. So this section are materials and methods. It's about the purification, concentration, and quantification of the DNA. So once we had chosen our samples and we had identified them, we had to purify the DNA from the fecal matter by using the Qui-Gon kit from Qui-Gon Inc. and following the manufacturer's instructions. By following the instructions, we washed away the impurities from the stool sample, leaving us with the isolated DNA. We then made the DNA, we made it 10 times more concentrated by using the Zymon kit from the Zymon Inc. And we followed the manufacturer's instructions by eluting our isolated DNA in the DNA elution buffer at a value that was 1/10 of our initial value. Once we had our concentrated DNA, we wanted to quantify the isolated DNA and estimate any contamination from proteins and RNA, measuring its purity. We found this out using the nanodrop spectrometer at 260 nanometers, and we used the ratio of absorbance at 260 nanometers over 280 nanometers to measure the purity of the DNA. We recorded the DNA concentration at the 260-280 ratio, and when making sure we had a high enough concentration of DNA extracted from the scat, we wanted to make sure that the concentration of DNA we had was purely DNA, not contaminated with proteins, because this would interfere with our results. We knew all the concentrations were high enough because we compared our 260-280 sample ratios to the acceptable pure DNA ratio range, which was 1.5 to 2.2. Sample 310 had initial DNA quantification of 92.8 nanograms and a 260 over 280 ratio of 1.8. Sample 518 had initial DNA quantification of 2.1 nanograms and a 260 over 280 ratio of 1.85. Sample 593 had initial DNA quantification of 92.7 nanograms and a 260 over 280 ratio of 2.07. And sample 863 had an initial DNA quantification of 1.9 nanograms with a 262 over 280 ratio of 1.5. All these ranges were within the pure DNA range of 1.5 to 2.2. So our next section is about the Amplicon PCR. After finding our DNA concentrations, we want to amplify certain variable regions of the prokaryotic 16S ribosomal RNA gene in our concentrated DNA. These were the V3 and V4 regions. In order to amplify these regions, we use the PCR amplification. So these are the reactions that we put together. In the positive control, we use 5 microliters of control DNA, which was E. coli. In the negative control, we didn't use any DNA. In sample 310, we used 1.1 of the extracted DNA. In sample 518, we used 4.7 of the microliters of the extracted DNA. In sample 593, we used 1.1 microliters of the extracted DNA. And in sample 863, we used 19 microliters of the extracted DNA. The target gene is first denatured by being heated to 95 degrees Celsius for 1 minute. It then goes through the annealing at 54 degrees Celsius for 45 seconds. Finally, is raised back up to the temperature of 72 degrees Celsius for 2 minutes for extension and DNA synthesis. These 3 steps were repeated for 30 to 40 cycles and created over 1 billion copies. The next section is gel electrophoresis. We use agarose gel electrophoresis to check the products of our PCR reactions and determine the size of the DNA fragments that we were analyzing. The smaller DNA fragments migrate further than the larger ones during the procedure because the distance it migrates is inversely proportional to the molecular weight. So on the chart we have our gel, and in the first lane was the negative control, which there was no band for. The next one was the positive control where a band was present. The next one was sample 310 where a band was also present. The next is sample 518 where a band was present, and sample 593 and sample 863, neither of them had bands. So we expected the size of the amplicon to be approximately 550 BP, and the presence of a band at 550 BP for the positive control lets us know that the DNA amplification worked. The absence of a band for the negative control is a good sign because the negative control to not have any DNA. The presence of a band for samples 310 and 518 shows us that the DNA amplification worked, but since there's no band for the samples 593 and 863, that means that the DNA amplification did not work. Our next section is about the PCR cleanup 1 and 2, the indexing PCR, the quantification of the indexing PCR products for the DNA sequencing. So we use the Asian Court and Pure kit, which is the PCR cleanup kit. We followed the manufacturer's instructions in order to clean up the amplicon from our amplification PCR reaction. This kit cleaned up by removing components of the amplification PCR buffer that included Mg, DNTPs, and enzymes. It also removed the forward and reverse primers that weren't used. We wanted to remove these molecules because if they remained with the amplicon, it would interfere with the indexing PCR by binding with the indexing PCR and primers. To perform the indexing PCR reaction, we placed our PCR tubes in the thermal cycle. First, it was set to 95 degrees Celsius for 3 minutes. Then it went through 8 cycles of 95 degrees Celsius for 30 seconds, 45 degrees Celsius for 30 seconds, and 72 degrees Celsius for 30 seconds. So after these cycles, we set it at 72 degrees Celsius for 5 minutes. Using an indexing PCR reaction, we were able to add a unique set of sequence tags to the end of the amplicon from each sample, which was done to help us identify our samples when we were sequencing the DNA. After indexing our samples, we performed a second PCR cleanup, this time using the Agincourt Ampere XP kit to clean up the amplicon from our indexing PCR reactions. This time, the cleanup removed the single-stranded DNA of the primers, DNTPs, buffers, and dyes so that it would not interfere with the DNA sequencing analysis. Once the amplicon was cleaned up, again, we used a fluorometric assay to determine the concentration of our amplified sample that we were left with after the cleanup. We used fluorescent dyes and measured the fluorescence using a cuvette fluorometer. So from our fluorometric quantitation readings, we determined our molar concentration of the DNA in our samples. Sample 310 had a molar concentration of 95.3 nanometers. Sample 518 had a molar concentration of 5.95 nanometers, and the positive control had a molar concentration of 16.4 nanometers. We used a fluorometric assay instead of the UV spectrophotometer assay that we used earlier because the fluorometric assay performs a more sensitive and selective and precise measurement, which will help us when we were preparing the dilutions for the DNA sequencing reaction. We took our fluorometric readings, which were measured in nanograms over microliters, and divided this number by 0.363 to determine our molar concentrations. As long as the concentrations could give us a final value of 100 microliters in a 4 nanometer solution, we were able to make our dilutions for the DNA sequencing. So here are our successful reads. We had to pick new animals due to the samples 310 and 518 not having analysis available, so now we have samples 671 and 672. The positive control had 8,036 successful reads. The negative control had 8 successful reads. Sample 671 had 6,315 successful reads, and sample 672 had 7,452 successful reads. All of them 100% passing the quality filter for us to base our analysis on, because the negative control barely had any reads and the other controls had a lot of reads. The positive control, out of the 8,036 successful reads, 8,035 of them were a type of bacteria. And at species level, the most abundant bacteria was E. coli alberti, making up 83.75% of the total reads, and the other 16.15% of the reads was a bacteria other than E. coli, which shows that there was a little cross-contamination among the reactions. So the unclassified category in the reads consists of reads that do not have any known organisms in that taxonomic level, while the percentage of unclassified reads increases as the taxonomic levels become more specific and get smaller and smaller. The reads labeled other are reads that are classified at a percent less than 3.5% of the total reads, so they're just labeled as other. Here are 3 charts showing our 3 different taxonomic levels: kingdom, order, and genus. At a kingdom level, all samples contain bacteria. At the order level, the positive control consists of enterobacteriales, and samples 671 and 672 contain bacterioides. At the genus level, the positive controls contain E. coli, and the samples 671 and 672 both contain Prevotella. So Prevotella has been found to cause periodontitis, which is an inflammation of the gums. This data was collected to answer our questions of whether similar weight would have an effect on the gut microbiome, and our hypothesis was that the gut microbiome would be the same, and our data does support this. Here are our sources.